Hello there everyone, and welcome to this Biology Mind A-Level tutorial on the structure of nucleotides. So in our last lesson we learned about enzymes and their function within the body and what factors alter the rate of enzyme controlled reactions. You now understand how the induced fit model is an improvement of the lock and key model, as well as the effects of inhibitors, drugs and metabolic poisons on enzymes. So our key learning objectives for today are all about our nucleotides. So number one, we're going to have a look at the structure of nucleotides. Number two, we're going to have a look at nitrogenous bases. And number three, we're going to explore what happens when we phosphorylate and dephosphorylate nucleotides. So let's get cracking. So first of all, let's think about nucleic acids. So DNA and RNA are both types of nucleic acid. Now, DNA stores genetic information, so it contains all the instructions for an organism's growth and all the instructions to execute necessary daily functions of that organism. Now, RNA has a very similar structure to DNA. As you can see, it has this helical structure. And it transfers the genetic information from DNA to help proteins form. So it's that interim messenger between DNA and final product of what DNA is there to do, protein manufacturing. So it's that DNA to RNA to protein formation chain that we need to think about. So that's our nucleic acids. Now, DNA and RNA are polymers made up of nucleotides. So nucleotides are the biological molecules that serve as the building blocks of nucleic acids, such as DNA and RNA. So let's have a think of what our nucleotide actually is. So you can see our nucleotide is formed from a phosphate group here, which we can also write as this. So this is our phosphate group. This is our pentose sugar. So it's a monosaccharide, and we know that it's a pentose sugar. Remember, pent for 5, because there are 5 carbons in its structure. And then there's a nitrogenous base. So this is what our basic structure of our nucleotides is like. Now, nitrogenous bases are organic, nitrogen-containing compounds. So this is our nitrogenous base on the end here, bound to our pentose sugar. And there are 5 different bases. So number 1, adenine, number 2, guanine, number 3, thymine, number 4, cytosine, and number 5, uracil. And our shorthand of noting these down is by just writing down the first letter of the word. So we say the bases are A, G, T, C, and U. What that means is that our 5 bases, either one of those is bound here to our pentose sugar. So this can be replaced by any one of these. Now we can split our bases into 2 different groups classified by their structure. So adenine and guanine are our purines. And then thymine, cytosine, and uracil are our pyrimidines. So all this means is that these have a similar structure and these have a similar structure. Okay, so our nitrogenous bases can be classified into either purines or pyrimidines. Now let's come and have a look at the difference in the structure between DNA and RNA. So you can see that this is our column for DNA, this is our column for RNA. And the first thing that differs between DNA and RNA is the type of sugar. So remember, we said that this is our pentose sugar. So they both contain pentose sugars, but they each have slightly different pentose sugars. So DNA contains a deoxyribose sugar, and remember that's in the name, deoxyribonucleic acid. and RNA contains a ribose sugar. So that's the first difference I want you to note. Now, the second difference is the nitrogenous bases between DNA and RNA. So DNA contains the bases adenine, thymine, cytosine, and guanine, whereas RNA contains adenine, cytosine, guanine, and in place of the thymine is a uracil base. So there's no thymine in RNA. That's a really important fact for you to remember. And equally, there's no uracil in DNA. Let's just summarise that quickly. So our key differences between DNA and RNA. RNA has a ribose sugar instead of a deoxyribose, and RNA has the base uracil instead of thymine. And make sure that you remember that DNA has thymine and RNA has uracil. That's a really important fact for you to remember. Really, really important, because this is a common mistake that many students make and that you will lose marks for in your exam. So DNA does not contain a base called uracil. Only RNA has that uracil base. And in place of the uracil base, DNA has thymine. Okay, so we have met nucleotides as a component of nucleic acids in DNA and RNA. So now let's meet them in their free form. So when a nucleotide gets a phosphate group added to it, it becomes phosphorylated. Now this is important with ADP and ATP, which I'm sure you've met before. So let's look at ADP. So ADP is a biological molecule consisting of the sugar ribose, which you can see here, and two phosphate groups, so 1, 2, and then a nitrogenous base, adenine. So this is ADP. So it's got two phosphate groups. Now let's contrast this to ATP. So ATP has the same sugar ribose, it has the same nitrogenous base, adenine. However, it's got an added phosphate group. So ATP has 3 phosphate groups. So ATP has 3 of these high energy phosphate bonds. And when one of these breaks, energy is released and ATP is converted to ATP. And then the ADP molecule in turn is converted back to ATP in a catabolic process in which energy was released that is used to make the high energy phosphate bond. So we need a catabolic process in which energy is released in order to add another phosphate bond to form ATP. And then in turn, when ATP loses its high energy phosphate bond to form ADP, it releases energy. It provides a source of energy. Okay, so note that when we go from ADP to ATP, we're phosphorylating. And when we go from ATP to ADP, we are dephosphorylating. We're taking away a phosphate group. And it's this taking away of a phosphate group that provides energy from ATP. Whereas, as you can see, from ADP to ATP, we need a reaction, a catabolic process, in which energy is released to phosphorylate our ADP. And some examples are given here, such as glycolysis, the TCA cycle, oxidative phosphorylation, and photosynthesis. Whereas here, when we're releasing energy by breaking our phosphate bond and forming ADP, we can release energy which can be used for cellular work and biosynthesis. So in today's tutorial, we've had a look at how nucleotides are a basic component of nucleic acids and explored the differences between DNA and RNA. Finally, we've come on to have a look at nucleotides themselves and how they can be phosphorylated and dephosphorylated, for example, with ADP and ADP. And there are other examples, but for now, just focus on ADP and ADP and how that in turn can release energy. So well done for completing this tutorial, and I'll see you for the next session.